Hi, hi everyone. I hope I'm audible. Let me just check once. Yes, okay. All right then. So let's get started. Okay. So ICSE Premier League. So that is match number 1. So Vani ma'am versus cell cycle and cell division. So I'm going to discuss about important questions that you can find. So this is about 3 marks or 5 marks questions, which you can get in your board exam, because previous year question papers that I have, previous year question papers, so from there I have added these questions. Okay, so let's have a look at that. And before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Just a second. Okay. Yeah. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. Okay. So this you can search in Telegram and you will find this channel. Okay. So you can join the channel for updates regarding the classes. So any class notes that are there like PDF, I can send it on my channel. Okay. And the Telegram channel, that is Unacademy, ICSE and ISC boards. So this link you will find in the description box. So you can go to the description box and find the link over there. Okay. So special classes you can learn from your favorite educator for free. That means we conduct special classes on Unacademy platform. Okay, which are absolutely free. Okay, and engage with educator via poll. So that means we will be conducting a quiz also. You can participate in those quizzes that we conduct. And at the end of this session, so we will be sharing the leaderboard so that you can know your rank as well. So clear your doubts in the live special class. So that is using the question tab. So there is a separate tab, so that we call a question tab, where you can type in your question, attach an image and send it to the educator during a live session, and they'll be clearing your doubt. So for the top special classes, you can visit the link in the description. Okay, now Unacademy subscription features, like what all what all you will have access to once you take the subscription. So one thing is basically you can learn live from the comfort of your home, unlimited access to all the courses. So different educators will come up with their own courses. Okay, so you will have access to all the courses once you take the subscription. Okay, then top educators of India are gonna take the class in one platform, that is Unacademy platform. Then there is regular doubt clearing session. Okay, so after about every 3 session, the 4th session will be doubt clearing session. So any doubts you have related to the topics that are previously covered, so you can clear your doubt in this session. Okay, then complete syllabus will be covered. Okay, then mentorship and guidance is given, study material is given. So we will also be providing the study material in the form of a PDF. Okay, the practice test, live test series, batch courses, you will have access to all of this once you take the subscription. Along with that, there is a daily practice section, then weekly mock test series, live quizzes, daily MCQ and subjective test series will also be there. Okay, and here you can have a look at, so since you are in class 9, class 10, okay, so you can try with 3 months of subscription. Okay, so now those who are planning to complete class 10 with Unacademy, so you can go with the 12 months of subscription. So this is, for 12 months it is around 15k with the referral code. So you need to add a referral code, that is Vani YT. So with this referral code you will be getting 10% discount on the subscription, so which will get reduced to somewhere around around 13.5k. Okay, so you can go for the subscription because you have many benefits. So basically you will have access to English batch, Hindi batch. So depending on your comfort, you can go for any of these batches. Okay, so let's have a look at some questions that I have added here. Okay, so all the questions that I have added here, so these are taken from Together With. Okay, so reference book here is the Together With. Okay, so you can also follow, so there is basically Oswald publication is there, then you have Goyal publication, so you can go with that. So for study and also textbook, you can anyway refer the this one, Selena. Okay, so Selena is the best textbook that you can refer for your ICSE class 10 board. Okay, for anyway for Together With, I mean for reference you can use the Together With. Okay, so let's have a look at this one. So state the differences between the following pairs. Okay, so mitosis and meiosis. One second, just a second. Okay, so state the differences between the following pairs. Okay, so what exactly might be the difference that you can find between a mitosis and then a meiosis. Okay, just a second. Okay, so I hope I'm audible. Yes, right? Yes, any of you know any differences between mitosis and meiosis that you can mention, where exactly mitosis occurs, where exactly meiosis occurs? Any idea? No? Okay, so let's have a look at this one. So mitosis is an equational division. So what is that equational division? If the parent cell, okay, so I'll just write it here in this. So let's say the parental cell is 2n. So let's say this is a parental cell. So it is having a 2 sets of chromosome. So now from this parent cell, whatever daughter cells that are produced, okay, so daughter cells which are produced, so those are going to be maintained as 2n itself. That is equational division. So why do we call it as the equational division? Here also you can find 2 sets of chromosome, and here also you can find 2 sets of chromosome. So that means here the DNA content was doubled, that is during the S phase of the cell cycle, and that DNA content is completely, it was doubled before the cell division, and that is equally divided. So only difference is that, so basically DNA was doubled here, and here DNA is equally divided. So that means here also you can find 2 sets of chromosome, and here also you can find 2 sets of chromosome, which we call as the equational division. Okay, so here now the mitosis, when we mention, so it is an equational division. So what is a meiosis then? So meiosis is a reductional division. So now this is with respect to mitosis. Okay, so I'll just tell you what exactly happens in meiosis. So in case of meiosis, let's see this. So now with respect to meiosis, all right, so meiosis is basically, it is a reductional division. So let's say parental cell is it. So now this is a parent cell. Okay, so this parent cell was how much? Like, it had 2 sets of chromosome. Okay, so parent cell had 2 sets of chromosome. Now we call this particular meiosis as a reductional division because this occurs in 2 stages. Okay, so that means, basically 1 set of the chromosome is only will be there in the gametes. So these daughter cells which are produced, so daughter cells will have the haploid set, that is only 1 set of chromosome that you can find in here. So this one is a reductional division. So you can see there is a reduction in the chromosome number. So this one is a reductional division. So why do we call it as a reductional division? Because you can see there is a reduction in the chromosome number, so which is getting reduced from 2n to n only. So that means here 2 sets of chromosome you can find, and here only 1 set. Okay, so 2 diploid daughter cells are formed. So whatever number of cells are formed here, so this is basically 2 cells you can find. Okay, so here 1 cell and here 1 cell, so 2 daughter cells. So this is 1 2, and this is the parental cell which was there. Okay, in case of meiosis, 4 daughter cells are produced. Okay, so it occurs in 2 stages. So here you can find 2 cells, and again this is going to produce 2 more cells. So this is, basically meiosis is gonna give us 4 cells. So 1 2 3 and 4. So this was 1 2, and here this was only 1. Okay, so that means 4 haploid cells are produced. So this was 2n, this is 2n, and this is 2n, that is 2 sets of chromosome. Here this was 2n, and this is n, this is n, and n will be maintained, that is haploid set of chromosome will be maintained in here. Okay, now only one division you can see. So this occurs only in one stage. Okay, so only one stage is there. Just a second. Okay, so only one stage, this occurs in only one stage, that is the first stage itself. Okay, so there are no further divisions over here, so only one division you can see. So this meiosis occurs in 2 stages. So there is this first stage, and then there is second stage of cell division that you can find in. Okay, just a second, let me just check it. Yeah, all right, so you can find it in 2 stages. Okay, now here in this case, so only one division is there. So now this occurs in a body cell. So what is that? Somatic cell. So somatic cell is all the body cell. So let's say there is a skin. Okay, so this skin you have damaged. So now new cell has to be formed. So now the older cells or a damaged cell which is there, so that has to be replaced. So now replacement of the old, or we can say the dead cell, so that is done by the mitotic division. So that is mitosis we can see, where the 2n, that is 2 sets of chromosomes, will be maintained as it is, and whatever new cells are formed, so they will also have the 2 sets of chromosomes. So this is in case of a body cell. Here in case of meiosis, it is it can be seen in reproductive cells. So that is sperm might be there, or ovum. Okay, so in those case we can see this meiosis. So that is basically the gametes. Okay, so gametes is where we can see the meiosis occurring, where the 46 chromosomes which were there in the earlier stage, so that will be reduced to 23 chromosomes. So instead of 46 chromosome, here you can find only one set of chromosome, that is 23 chromosome. Here you can find 23 chromosome. Okay, so when the sperm and then the ovum combine, so 23 will combine. Okay, so that means whatever zygote is formed now, so this zygote is going to have the 46 chromosome. That means the normal number is maintained in here. Okay, so now this is the meiosis you can see here, as the 2n is reduced to n, so that is in meiosis. So that is because of the combination which can gonna occur, which is fertilization is gonna occur later on, that is the fusion of the male gamete and the female gamete, so that the normal chromosomal number is retained in an organism, and that is the reason the 2n is getting reduced to n in a meiosis. Okay, so instead of 2 sets of chromosome, only 1 set of a chromosome that you can find. Okay, so let's have a look at the next question. So the next question is basically the diagram based question. Okay, so this is a diagram based question, so you can have a look at this diagram which is mentioned, and this question was asked in the year 2017. So this is taken from the book Together With. Okay, and so this is basically for ICSE class 10 itself. So let's have a look at this one now. All right, So now study the diagram given below which represents a stage during the mitotic cell division. So this is mitosis itself, and answer the questions that follow. Okay, so what is the question? So let's have a look at the different questions which are asked here. Identify the stage giving suitable reasons. Any idea what might be this stage? Any of you? What is this stage now? So study the diagram given below which represents a stage during the mitotic cell division and answer the questions that follow. So this stage you need to find out. So you need to make an observation with respect to furrow formation. You need to make an observation with respect to nuclear membrane reappearance. You need to make an observation with respect to nucleolus reappearance. So based on that, you can say which stage is this one. Okay, name the part numbered 1 and 2. So what is this one here? Okay, so what is this part and what is this part? Okay, so this you need to know. All right, then what is the technical term for the division of the nucleus? Any of you can type in your answer, so I'll just check it here. Okay, so any idea you guys have, you can just type in. I hope I'm audible. Just a second. All right. Okay. So now what is the technical term for the division of nucleus? Then mention the stage that comes before the stage shown in here. So like you need to mention which stage comes before this stage and draw a neat label diagram of the stage that is mentioned here. Okay, so which is the cell division that results in half the number of chromosomes in a daughter cell. So let's see this one. So basically this phase what you see now. So answer for this one, identify the stage giving suitable reason. So what is this stage? So this stage is basically a telophase. Okay, so this is telophase. And in telophase what you can observe, so basically nuclear membrane is gonna reappear. Okay, so nuclear membrane reappearance you can see. The nucleolus is gonna reappear, so you can see the nucleolus also so that means this is totally opposite to that of a prophase. In prophase what exactly happens? The nuclear membrane disappears, the nucleolus disappears, whereas here in this one, so basically the telophase, when we mention nuclear membrane reappearance we can see along with the nucleolus. So name the parts numbered 1 and 2. So what are those parts numbered 1 and 2? So you can have, okay, so reason I did not mention. So yes, I already mentioned. So two sets of daughter chromosomes are seen in the opposite pole. So that is their reappearance of the nuclear membrane and nucleoli. Okay, so formation of the chromatin fiber and deepening of the cleavage furrow. So this is this formation you can see. So there is a furrow formation that you can see, that is at the end of the late anaphase only you can see this one. Okay, but anyway here you can also see that the chromatin fibers are being formed. Okay, so chromatin fiber is being formed, nuclear membrane is there. So now the chromosomes are shown and nuclear membrane. So this is basically still a chromosome, but actually chromatin fiber is formed. So you can just take it as a chromatin fiber itself. So because you can see a chromatin fiber formation that is at the late telophase. Early telophase if we can see, so yes, there is a chromosome itself. Okay, so based on that now, so nuclear membrane is here, the next one disappearing in the prophase. Here it has reappeared. So this second part is basically a nuclear membrane. Okay, so this one is a nuclear membrane. So let's just write that. Yeah, So now this part is nuclear membrane. So this one is chromatin fiber depending on early stage or late stage. So this is going to be chromosome. Okay. So here still they have shown a chromosome itself. So they have not shown a chromatin fiber yet. Okay. So next question is what is the technical term for the division of the nucleus? So what is that technical term for the division of nucleus? So karyokinesis. So karyokinesis when we mention, so basically division of nucleus you can find. So that means prokaryote, eukaryote, you have heard these terms, right? So prokaryote, eukaryote when we mention, the karyote is basically a nucleus. Pro is primitive type of nucleus, U is true type of nucleus is there. So now you know karyote is basically a nucleus. Kinesis is division of the nucleus. The technical term is basically a karyokinesis. Okay. So mention the stage that comes before the stage shown in the diagram. So draw a neat label diagram mentioned. So you can draw the anaphase here. So I'll just show you this anaphase structure here. So you can see the daughter chromosomes are being separated, then like aster rays formation you can see. So aster rays are this one. Okay. So aster rays are there. Okay, which will together we can call. So now you know this is a centriole. Okay. So surrounding centriole you can see this aster rays. Okay. So now I'll just label this as centrioles are there. Okay. And along with the centriole you can see a aster rays there. Okay. So now aster rays and centriole together, so we call it as a aster. So aster formation we can see here in the prophase itself. So now these asters are there. So aster rays you can see. So centriole is there and spindle fiber formation. So what exactly has happened here? The chromatids are separated. Okay, so these chromatids over here are separated. So this is a daughter chromosome we can mention. All right. So next, what is the question? Which is a cell division that results in half the number of chromosome? That means 2n is getting reduced to n. So which we call as a reductional division. So which one is a reductional division? Then the reductional division is basically a meiosis. Okay, so meiosis, that means instead of two sets of chromosome, the daughter cell will have a single set of a chromosome. Okay, so let's have a look at the next question now. And this question was asked in the year 2016. Okay, so this is from the year 2016, and again these questions are taken from the together with. Okay, so this I have taken from the together with itself. So let's have a look at this one. So the given diagram shows a stage during mitotic division in an animal cell. Okay, so in an animal cell the diagram is shown that is stage during a mitotic division. So identify this stage. So what is this particular stage now? Okay, so identify the stage, give a reason to support your answer. So each and every division that you need to know. Okay, so now draw a neat label diagram of the cell as it would appear in the next stage. Okay, so draw a neat label diagram. So you need to show the next label diagram of the cell. It would appear in the next stage and name the stage. So you need to name that stage and you need to draw that stage. Okay, so let's have a look at the other questions and then we'll discuss the answers. Okay, so in what two ways is mitotic division in an animal cell different from the mitotic division in an plant cell? Okay, so now how exactly mitosis is different in plant and animal cell? So that is asked. Now name the type of cell division that occurs during the growth of a shoot. Okay, and formation of the pollen grains. You need to analyze here because the shoot is with respect to the plant body. Okay, so this is with respect to plant body formation, and formation of pollen grain is with respect to what? That is with respect to gamete formation. So based on that you can mention whether this is a mitotic division or a meiotic division. Okay, so now given diagram shows a stage during the mitotic division in an animal cell. So identify this stage. So what stage is this? So what exactly happens in prophase? Any of you? So this is basically a prophase itself. Okay, so this is a prophase. Reason being the nuclear membrane is gonna disappear. So you can see that the nuclear membrane is disappearing. Okay, and also you can see that the chromosomes are formed. So let me just erase this part, just a second. Yeah, so now you can see in this. Okay, so chromosomes are being formed here. So what is this chromosome? Basically the chromatin fiber is condensing and chromosome formation that you can see here. Okay, so identify the stage, give reason to support your answer. So chromosome formation you can see. Now draw a neat label diagram of the cell as it would appear in the next stage. So like what exactly would be the next stage here? Okay, so in that case, one second. Okay, yeah, so next stage after the prophase. So what is the order? Like, first the prophase is there, then there is metaphase, where all the cells, I mean all the chromosomes are going to arrange at the equator. Then there is anaphase, where these chromatids are going to move towards the pole. Then there is telophase, the deepening of the furrow. You can see nuclear membrane, nucleolus, so they are going to reappear. Okay, so in that case, in what two ways is mitotic division? So now next question is, draw a neat labeled diagram of the cell as it would appear in the next stage. So what is the next stage? And you need to draw that and you need to name it. So what is that? The next stage is basically a metaphase. Okay, so metaphase, you can see all the chromosomes are arranged here in the equator. Okay, so duplicated chromosomes you can find. The aster is there as usual. The centriole is there. So spindle fiber you can see, and then you can also see there is a chromatid. Okay, so chromatid formation also you can see. Okay, so now this stage is basically is drawn, and along with that the metaphase is shown. Okay, in what two ways is mitotic division in an animal cell different from the mitotic division in a plant cell? So this is in comparison, like how exactly mitotic division in animal and plant cell is different. Okay, so let's have a look at this one, the mitosis in plants and mitosis in animals. Okay, so how exactly are they different? So now in case of plants, so asters are not formed due to lack of centrioles. So there is no aster formation. What is aster formation? So centriole will be there. So surrounding centriole you can find, so there is a aster ray. Okay, so this is a aster ray. So let me just label this one. So this is now a aster rays. Okay, and these are centrioles. So together we call them as what? So together we call them as a aster. Okay, so in general it is a aster. So aster formation you do not see in case of plant cell, because there is no centriole, because there is no centrosome. Okay, so here the asters are formed. So you can clearly see this is an animal cell now, because there is no cell wall. So you can see there is a aster formation. Now formation of cell plate you can see in the telophase. So what exactly is the case here? Now so there is a cell wall. Okay, so let's see, here is the cell wall. Okay, and inside the cell wall you can also find there is a cell membrane. Okay, so there is a cell membrane, and you can see there is a cell plate formation. One second. Okay, so there is a cell plate formation that you can see. So where exactly this occurs? The cell plate formation going from where to where? This is growing from the center, that is from inside to the outside. So this will start growing, and it is going to separate the cell wall also. Okay, so cell wall and cell membrane are separated. So now we get this as one cell. Okay, so now this is one cell that you can find, and this is another cell that you can find. Okay, so these are the two cells which you find. So in that case, formation of cell plate you can find in a telophase. A constriction forms in the cytoplasm, so that is in the telophase. So what is that constriction? So if it is an animal cell, so we would see that this is an animal cell now. Okay, so constriction is basically a furrow formation that you can see, which will grow from outside to the inside. So deepening of this constriction you can see. So now this furrow is going to separate, like it is going from outside to inside. So you can see there are two cells that are formed. Okay, so these are the two cells that are formed there. Okay, so a constriction forms in the cytoplasm, so that is in the telophase. Is this clear? Any doubt in this? Okay, so now the next question is, the diagram given below represents a certain stage of mitosis. Okay, so a particular stage of mitosis is mentioned here. Okay, so identify the stage of cell division. So what is the stage of cell division? And you need to know what is this stage now. Name the parts labeled A and B. So what is A and what is B? So you can see the A is labeled here, the B is labeled here. Okay, so what is A and B? Then what is the unique feature observed in this stage? So what is that unique feature which you can observe in this particular stage? Okay, and how many daughter cells are formed from this type of cell division? So you can see there is a cell division that is occurring. Okay, so how many daughter cells are formed from this type of a cell division, which you can see here? So now this, the diagram identify the stage of cell division. Any idea which is this stage number? Yeah, so this stage is basically a anaphase. Okay, so this one is anaphase. So you can see that the daughter chromosome, like you can see these chromatids are separated. Okay, so double the amount of chromosome. Earlier it was half. So earlier these were the chromatids, like attached at the centromere. You could see this chromosome. Okay, so now there is a division that you can see. So because of that division, so basically centromere splitting you can see. So two of the chromosomes, like the chromosomes are split up. I mean chromatids are split up along with the centromere splitting is occurring. So this is basically due to contraction in the spindle fiber. So there is a force that is created. Okay, so spindle fiber you can see between them. So this one is a spindle fiber here, and these are the chromatids. So identify the cell division. So this cell division or stage is basically a anaphase. Okay, so next one here, this one is a spindle fiber, and the B is basically, what is it? That is a centromere. So you can see this part, so they are specifically pointing these parts now. So this is a centromere. Okay, so what is the unique feature observed in this stage? So what is that unique feature that you observe in this stage? Any idea? No? So unique features, so sister chromatids of each chromosomes are separated and move towards the opposite pole. So that is because of the contraction in the spindle fiber, the force that is acting on this for the separation. So that is basically contraction of the spindle fiber. which is making the sister chromatids to separate. So that means the sister chromatids are moving towards the pole. So there is the, the movement is occurring towards the pole now. So how many daughter cells are formed from this type of cells, cell division? So that is 2 daughter cells are formed here. Okay, so 2 daughter cells, that is because it is a mitotic division, since it is not mentioned about anaphase 1, telophase 1, so whether it is occurring in stages. So this is gonna give you 2 of the cells. So that means the parental cell is there, which is getting divided into 2 daughter cells. So that is 2 daughter cells are formed here. So this is gonna be 1 cell, and this is gonna be 1 cell. So with the cell wall as as usual. So this is a mitotic cell division. So this is mitosis. Hi Ankit, I'm good. How are you? Alright, so let's have a look at the next question now. So this is about, so yeah, so this is the next question. So this is from the previous question itself. Okay, so given below is a diagram, so you can have a look at this one. So this question was asked in the year 2014. Okay, so this is from the year 2014. So given below is a diagram representing a stage during mitotic cell division. Okay, so study it carefully and answer the questions that follow. Okay, so you need to have a look at these diagrams that are mentioned in here. Just a second. Yeah, and you need to study the questions that are followed. So this is a diagram based question again. So these are basically, you can have these marks for have these kind of questions for 5 marks, or maybe for 3 marks as well. So some questions they'll reduce. So certain cases it can be for 5 marks and it can be for 3 marks as well. Okay, so now is it a plant cell or an animal cell? So how are you going to understand whether this one is plant cell or an animal cell? Any idea? So how do you differentiate this one? So whether this one is plant cell or animal cell? So this one is basically a plant cell. So how are you going to understand that? So that is based on the cell wall presence. Okay, so since the cell wall is present here, okay, so we can say that this is a plant cell. Okay, so give a reason to support your answer. So you can say cell wall is present as one of the reason, and there are no asters. Okay, so aster formation you do not see in case of a plant cell because centriole is not there, so there is no aster formation. So based on that we can see this is a plant cell now. Okay, now identify the stage below. So what is this stage? So whether this is a prophase or metaphase, anaphase, telophase. So this is the first stage itself, that is the prophase. Okay, so prophase, so centriole is not present, it is having a cell wall. The next one is basically a prophase because you can see the nuclear membrane is disappearing. Okay, nucleolus has already disappeared. The chromatin fiber has condensed to form a chromosome, and there is a perfect spindle fiber formation. Okay, so now this one is basically the plant cell. So this I gave you the answer. So this one is prophase. Okay, the next one is name the stage that follows the one shown here. So how is that stage identified? So next stage is basically a metaphase. Okay, so what exactly happens in metaphase now? So in metaphase the chromosomes are arranging themselves at the equator region. So which is that equator region? So you can see here. So I'll just highlight that. Just a second. So now all the chromosomes will get arranged here. Okay, so that is at the equator region. So next stage would be the metaphase. Okay, and as all all of them are getting arranged where? So they are all getting arranged at the equator. Okay, so how will you differentiate between the mitosis and meiosis? On the basis of chromosome number in the daughter cell. So this is with respect to mitosis and meiosis. So in mitosis we will have the 2n only. So 2n, that is two sets of chromosomes, are maintained as two sets of chromosomes only. Here in this case, so only one set of chromosome that you can find. So that means two sets of chromosome is getting reduced to one set of chromosome. Okay, so that means there is a reduction that you can see. So that means this is a equational division. Okay, so because same chromosomal number is maintained, and this one is a reductional division. Okay, so this is a reductional division. So you can have a look at these ones. So because here you can see only one set, so 23 chromosomes are there, and here you can see 46 chromosome. That means two sets of chromosome that you can find. Okay, so now draw a duplicated chromosome and label its parts. Okay, so what is that duplicated chromosome? Now during a cell cycle, so I'll just explain it to you here. Okay, so now this one, let's say cell cycle is there. Okay, so this is a cell cycle. So in cell cycle, so you can find a M phase. Okay, so there is a M phase. So this is completely a M phase, and the rest of this, like this whole thing which is there basically, so this is interphase. Okay, so this one is interphase now. All right, so what exactly happens in G1 phase? In G1 phase you can see that different changes. So basically raw materials are kept ready for the S phase, and here is this S phase where the DNA is gonna replicate. So DNA replication you can find in here. So with respect to that, so draw a duplicated chromosome. So instead of two arms of a chromosome like this, okay, one second, so I'll just show you there itself. Yeah, so let's see here itself. Okay, so in this case what would be the condition now? So here you can see two chromosome, like two chromatids are formed. So instead of one, why? Because so now we have the double the amount of chromosome, as in the S phase the DNA replication is occurring. So we are not showing the chromosome like this now. Okay, so we are not showing the chromosome like this. We will be showing the chromosome like this. That is the chromosome with not just with two chromatids. So we are showing a sister chromatids here. So four of the chromatids we are showing, because in S phase what happened? In S phase of cell cycle, the DNA replication happened. So whatever previous amount of DNA was there in this chromosome, so that has completely got doubled. So you can see there is a sister chromatids formation. So centromere is there and the chromatids are being formed. Okay, so chromatids are there, so chromatid formation. So this is double the amount, because this is completely, here you can find a DNA over there. Okay, so this is completely a DNA there. Just a second. Yeah, so double the amount of DNA you can find in this stage. All right, one second, so I am not able to see this cursor. date is May 11th. Okay, so and the mode of delivery is English. So here also the same rule is applicable and PCMB is covered. And introducing community, so never miss on any update from your favorite educator. So get session notes and practice material. Share your feedback directly with the educator via poll. So link you will find in the description. Okay, so opportunity for all learners to report any inappropriate content in the video. Okay, so be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Okay, so how you can watch my special classes? All you need to do is download the Unacademy Learner app. Open the app and click on get started. Then enter your mobile number and OTP. So sign up using your email ID. Okay, so then go to the description of this video. So basically you need to select your class, also select your category, so that is ICSE category. Then you can select the preferred language that you would like to learn in, and after that click on the link, then click on notify me, and then you can use this code Vani YT to unlock many of the free products that are there on Unacademy platform. Okay, so see you all in the next session. So thank you everyone. So subscribe to our channel, that is Unacademy ICSE and ISC boards. Click on bell icon to get notified about the classes. Like and share the video. Download the Unacademy Learners app now. The PDF anyway I can send it on my Telegram. Okay, so see you all in the next session. Bye bye everyone. Thank you so much.